Hello students, we start with the 4th semester syllabus of Unit 1 of Animals and Cell Culture. Unit 1 in Animal Cell Culture part, it deals with the introduction to Animal Cell Culture. Maybe that is the 4th unit in the syllabus, okay, but the first unit in Animal Cell Culture part, we start with the introduction to Animal Cell Culture technology. So first we'll define what is this animal tissue culture or animal cell culture. It is defined as an in vitro cultivation of organs. An in vitro cultivation of organs, tissues, cells at a defined temperature using an incubator or supplemented with medium containing cell nutrients and growth factor is collectively known as tissue culture. So, in vitro cultivation of animal organs, cells, tissues at a defined temperature using an incubator and supplemented with a media containing cell nutrients and growth factors is collectively known as animal tissue culture, and it will continue to grow if supplied with appropriate conditions and nutrients. That means you can grow them and you can maintain the cells in a homogeneous kind of cell types in the form of cell lines, okay? And this culture process allows the single process to act as an independent unit, and the cells are capable of dividing and growth in vitro condition. So this is how it is carried out in aseptic condition in the animal cell culture laboratory. Introduction to this, it has become one of the major tools to be used in life science industries today, and also for study purpose, for research. The tissue culture is one of the general term for the removal of cells, tissues and organs from an animal or a plant and other subsequent placement into an artificial environment conducive to growth. So this environment usually consists of a suitable glass or a plastic supported culture vessel containing a liquid or semi-solid media that supplies the nutrients which are essential for survival and growth. And you have to remember that in vitro growth, they require the same environment as in vivo. In vitro culture, maintenance and proliferation of cells, tissues or organs. So these are the different types. Animal cell culture can be carried out for organ culture, for a growth of specific organs, tissue culture, specific tissues, cell culture, histotypic culture, organotypic culture, primary culture or cell line. Okay. Now these are the types of tissue culture. So what are the characteristics of this animal cell culture? Animal cells can grow in a simple glass or a plastic container in a nutritive media, but they grow only in a limited generation. The normal cells, they grow, they have their lifetime where they follow this normal growth curve, okay? Lag phase, log phase and decline phase. The normal cells, and they undergo death after their lifespan. The maintenance of growth of cells under laboratory condition in a suitable culture media is known as primary cell culture. Primary cell culture, if you take the tissue directly from the organism and grow them, maintain them, we call it as a primary cell culture. If you subculture it, it is called passaging or subculturing. The cells can grow as the adherent cells. This is the characteristic feature of animal cells where they are anchorage dependent. That means they need some support for their growth. That is, they need an anchorage. That means usually it is provided by the fibrin or proteins which are provide anchorage for the growth of these animal cells. Must be cultured while attached to a solid or semi-solid substrate. And in the suspension culture, anchorage independent cells can be grown floating in the culture media. So anchorage dependent cells like blood cells where they move, okay, they grow floating in the culture media. But all the other cells, the animal cells, they show the nature of anchorage dependence. So if you look at the history of animal cell culture development, you have to remember long list is there. First, we always remember Harrison in 1907. He is also considered as the father of animal cell culture technology. He was the one to grow the frog embryo nerve fiber outgrowth in vitro. He cultivated frog nerve cells in lymph clot held by the hanging drop method. Hanging drop method is that, so he used a slide with a depression, okay, and he sealed the cells which are grown in vitro, and the growth of the cell in the depression, it is appeared as a drop where the cells were multiplied and grow. So that was appear as a hanging drop. So that method was called as hanging drop method, and observed the growth of nerve fibers in vitro. In 1912, so you have to remember a person, Alexis Carrel. He was the one which grown the chick embryo connective tissue in the heart muscles, the contractile cells for 2 to 3 months in vitro. Explants of chick connective tissue. In 1916, Ross and Jones, they conducted trypsinization and subculture of explants. In 1920 and 30s, it is Carrel, he subcultured the fibroblast cell lines. In 1925-26, it is differentiation into in vitro in organ culture was done. 1940s, that is introduction of the use of antibiotics in cell culture media, for that is the antibiotics which are used here, penicillin and streptomycin, for avoid contamination. Next, we have to remember early, that is establishment of L cell mouse fibroblast cell lines. Okay, first continuous cell lines are obtained in 1943, 1949. The growth of virus in cell culture was done. In 1942, use of trypsin for generation of replica subculture. Virus plaqueosae was done. 1952, gay establishment of first human cell line, HeLa, from the cervical carcinoma cells. That is the name of a person where the cervical cancer cells are obtained. That's why they are named as HeLa. In the Eagle 1955, development of chemically defined media with all nutritive components. 1959, Cook and Morris, cloning of HeLa and homologous feeder layer. 1961, the cell fusion of somatic cell hybridization was done by Suriol and Amphosi. And this is history and developments. 1962, Macpherson and Stroker establishment of transformation of baby hamster kidney cells. That is 21 is their series. And Klein, Smith and Pearson, the pluripotency of embryonal stem cells was done. 1965, Ham, the serum-free cloning of Chinese hamster cell was done. And Huber and Cohen, the epidermal growth factor was determined. 1968, Stroker et al. Anchorage independent cell proliferation was done. And in 1969, Metcalf, a colony formation of hematopoietic cells was done. Kohler and Milstrain, hybridoma technology for the production of monoclonal antibodies was done. In 1976, totipotency of embryonal stem cells was also done. And 1976, Hyansi and Sato, growth factor supplemented with serum-free media. And 1977, Nelson, Rees and Flanders Mayer was confirmation of HeLa cell cross contamination was done for contamination of many cell lines. Confirmation of HeLa cell cross was done. In 1978, selective serum-free media was developed. In 1980 and 87, development of many specialized cell lines was established. 1982, Dornell regulation of gene expression. Evans in 1983, regulation of cell cycle. 1984, Colin, production of recombinant tissue type plasminogen activator in mammalian cells. In 1987, Weinberg, oncogenesis, malignancy and transformation. In 1990, development of the laminar airflow cabinets for animal cell culture techniques was done by Collins and Kennedy. 1991, Griffith, polio vaccine grown in monkey kidney cells in vitro. And 1998, Thomson et al. Culture of human embryonic stem cells was done. In 2000, the Human Genome Project, genomics, proteomics and genetic deficiencies and expression errors were determined by the completion of the Human Genome Project. Major research in the animal cell culture in 2001, it is characterized the composition and function of adhesion in three-dimensional 3D culture was developed by Edna et al. And Nicola et al. In 2001, enhancement of hepatitis C virus RNA replication by cell culture adaptive mutation. And Annie Sittler et al. In 2001, they demonstrated the Huntingtin protein aggregation in cells can be suppressed by chemical compounds activating the specific heat shock response. The major research in 2003, it is long-term polymerization of culture and germline transmission of mouse male germline stem cells. In the long-term spermatogonial stem cell culture was useful in studying the spermatogenesis mechanism, has an important implication for the developing new technology in transgenesis and medicine. And in 2003, the integration of the cell culture and microfabrication technology and the recent progress in cell culture and microfabrication technology has contributed to the development of cell-based biosensors for functional characterization and detection of drugs, pathogens, toxicants and allergens. And in 2003, the one more development was that is Umero, a transplantation of culture bone cells was using combination of scaffolds and culture technique was developed. There's a step towards the bone regeneration therapies. The major research in animal cell culture in 2005, Bolviolik and et al., the microfluidic platform for complete mammalian cell culture and automated cell microculture technique was developed. And Wang and et al. In 2005, a bioreactor, a powerful tool for large culture of animal cells for extraction of therapeutic proteins was done. Ako and et al. Laser guided direct writing of three-dimensional tissue engineering technique was developed, and they did for liver and pancreatic morphogenesis and differentiation in angiogenesis. Angiogenesis is the formation of new blood vessels. Exo et al. In 2005, inkjet printing of viable mammalian cells was done. 3D printing, the computer-aided inkjet printing of viable mammalian cells holds the potential for creating a living tissue analogues and may eventually lead to the construction of engineered human organs. In 2010, 3D cell culture was developed, and Ang B, they did the microfluidic cell culture in controlled microenvironment. Ugan and et al. In organ re-engineering through the development of transplantable recellularized liver graft using recellularized liver matrix. And the design of pre-vascularized three-dimensional cell dense tissue was also done by the Sasagawa and et al. In 2010. And you have to remember this protein called CRISPR-Cas9. It is the first discovered by Emanuele and Chad Partner and Jennifer D. and awarded the Nobel Prize in Chemistry for 2020 for discovering the one gene technology sharpest tools called CRISPR-Cas9. It is called cluster regularly interspaced short palindromic repeats is also called CRISPR associated protein 9. That's why it is called as CRISPR-Cas9. It is used to edit the genes. The researchers can use these change DNA of animals, plants and microorganisms with extremely high precision. In April 2015, a Chinese group reported that first application of CRISPR-Cas9 for gene editing on non-viable human embryos. So, because of ethical question, it is still not applied for human embryos. This development, together with the decreasing cost of technology, have triggered a major bioethical debate over how far this technology should be used on living organisms. This is about the development of the animal cell culture from 1907 till 2020, and how these cell culture techniques are used, where they are applied. They are applied for the production of pharmaceutical and medical application. It is also called red biotechnology. The products of this animal cell culture can be obtained for therapeutic application. They are called as therapeutic proteins, example glycoproteins, antibodies, etc. Antibodies can be used for diagnostics and development of diagnostic kits, the production of viral vaccines. The cell products and regenerative medicines, they are also used in cell therapy for bone marrow cells, stem cells, and functional tissue transplantation and tissue engineering purpose. And extracorporeal organoids are also for artificial synthesis of artificial liver and kidney. The cell culture can be also used for substance testing, in vitro testing for replacement of animal trials. Okay, so where the model organisms can be replaced with in vitro culture of these animal cells, and drug testing and substance validation of the drugs, toxicity and chemicals. Okay, so what are the advantage and disadvantage of doing this animal cell culture? Advantage is the development of cell line over several generations. The scale up is also possible, and absolute control of physical environment where it is not possible in the complex organism, and homogeneity of a sample, the less compound needed than the animal models. Disadvantage is that cell may lose some differentiated characteristics, hard to maintain, only go small amount of tissues at high cost, and de-differentiation and genetic instability and aneuploidy. See, these are the disadvantages. So I have given you the brief introduction to animal cell culture technology. Just watch this video.
Successful cell culture depends upon many factors, including the quality of the cells and reagents you use, your sterile technique, the amount of experience you have, and the right laboratory equipment. Proper setup is important. Have everything you need close by. The basic equipment you'll find in a cell culture lab includes a cell culture hood, an incubator, a water bath, a centrifuge, a refrigerator freezer, a hemocytometer, and a microscope. You'll also need cell culture flasks, medium and other reagents, pipettes, and waste containers. The hood provides a sterile environment to work with your cells without fear of contamination. Always open your cell culture dishes and reagent bottles in a sterile environment. Outside the cell culture hood, keep a stack of disposable pipettes nearby. Pipettes are placed in the hood during use and only opened inside the sterile field. Have the trash, biohazard bin, and sharps container close by. A cell counting machine, such as the Countess, gives you quick, accurate counts needed during culturing. Or you can count cells manually with a hemocytometer. You may also wish to have the centrifuge located in the cell culture facility. Incubators provide the appropriate environment for cell growth. Carbon dioxide and temperature settings depend on cell type and medium selection. Mammalian cells are cultured at 37 degrees Celsius in the presence of 5 to 7% carbon dioxide with high humidity. Inside the incubator, keep your cell culture flasks evenly spaced and avoid crowding to ensure that the air inside the incubator moves freely. This helps to maintain temperature, humidity, and proper gas exchange. Don't make very tall stacks of plates. They may fall over, and lower ones may not get the appropriate gas exchange. Store all media and reagents according to the instructions on the label. Some components are known to degrade when left at room temperature or exposed to light for extended times. If the medium will be stored in a glass front refrigerator or a cold room, wrap the bottles in tinfoil or store them inside a box. Some labs warm their reagents to 37 degrees Celsius before use. Warming reagents should take 10 to 20 minutes, depending upon the size of bottles you are using. Others choose to use reagents warmed to room temperature or directly from 4 degrees storage. If you use a water bath, make sure to keep bottles upright, with necks out of the water. Specially designed bottle weights are available for use with the Gibco bottle. Alternatively, metallic bath beads can be used instead of water. Bath beads are a cleaner, greener alternative to the water bath. While waiting for reagents to warm, prepare the materials you'll need to use. When working under the hood, make sure the hood sash is at the proper height. Correct chair height and body position prevent shoulder stress and also helps prevent contamination. Never breathe or talk into the hood. Wipe the hood with 70% ethanol before and after use. Each item in the hood should be sprayed with 70% ethanol and wiped clean. Items within the hood are usually placed as follows: pipette aid in front, reagents in back, tube rack in the rear left, small container to hold liquid waste in the back right, your cell plates in the center. Never leave reagents in the water bath longer than necessary. Excessive warming can lead to L-glutamine degradation and loss of activity in trypsin solutions. Gipco Glutamax and Triple are more stable versions of L-glutamine and trypsin. Remember to clean each item placed in the hood with ethanol. You may choose to spray the item before placing it in the hood or immediately after, as demonstrated. Transport your cells carefully. Don't allow damage to the cell monolayer and avoid medium splashing that can encourage contamination. If using flasks with unvented caps, be sure to tighten before removing from the incubator. View every culture under the microscope daily. Check on cell health and growth and look for contamination. Make sure you're working with the cells you are expecting in your culture. If there's any doubt about cell identity, get a new culture from a reputable source or use a cell authentication kit.
Okay, this is available in the YouTube also, you can watch. Thank you for watching, keep watching.